So after moles and after bats, now we move to fish. And I'm very happy to welcome Joost Woltering. Joost comes from Konstanz, the University of Konstanz in Germany. And he's going to tell us about beyond adaptation, how genetic architecture shapes the fish dermal skeleton.
Many thanks, Denis. First of all, it's of course a great honor to be able to give a lecture at the Collège de France. So on my first slide, you see a fish skeleton that is labeled for calcium staining. So you basically look here at the dermal skeleton of the fish. And this is a quite extraordinary fish. It did something that few other fishes actually did, because, you know, fishes, they have, you know, a skin, and of course on the skin there is something to cover it, and in most fishes, lots of scales. This fish got rid of its scales and then got something for it in place, namely bony plates. And this is the structure that I will come to. The second part of the talk, first to start with, I would like to share some more general reflections on evolutionary theory and the kind of questions that we try to answer, and how we, and I think this is important, how we try to interpret natural diversity and natural differently occurring phenotypes. Of course, the main agenda of evolutionary biology is to explain the patterns and diversity of life. Darwin's endless forms, most beautiful, most wonderful, you all heard it. Life is diverse. Evolutionary theory tries to understand and explain by which mechanisms, by which processes in a historical context we came to diversity as it is now. And you all heard about that, of course. The primary paradigm that is being used is Darwin's descent with modification, how over the eons, how over the billions of years, life, you know, transformed from a single-celled organism into the multitude, probably even into the hundreds of millions of different species we know today. But there are quite different ways in how different evolutionary researchers put the emphasis on trying to understand this process. And to make this clear, and to really, you know, point out where we, or where I am standing in my research and which kind of questions I try to understand. I here put the different evolutionary the different parts of the evolutionary process sort of conceptualized into different boxes. Let's say the flowchart of evolution. So in the first box here, we have heritable natural variation. Of course, populations of one species, they're not genetically homogeneous. Different species, they vary different, you know, very slightly in their genetic makeup. They mate with each other, and the differences in genetic makeup will inevitably, you know, influence how successful they are in reproduction, and how those different genetic variants get transferred to the next generation. Different genetic variants will have a different rate of transmission. And this, of course, is the process of natural selection. If animals are better, you know, adapted to their environment, if they have better solutions, genetic solutions to their environment, they will reproduce more. And then these kind of genetic variants will be amplified in the population. And of course, this process of natural selection is the agent of Darwin's theory of evolution. And most of the time, what we see in this box, basically this is the box where the morphology ends up. We interpret this morphology as a solution to the environment, as adaptive change. And you all have recognized, of course, this process as survival of the fittest. The better adapted animals, they reproduce more, they get more, you know, they get more numerous, those genetic variants. And this arguably is the part of evolutionary theory that has been most focused on during the crafting of what is called the modern synthesis, the modern evolutionary theory during the first and second half of the 20th century. What is here in the first box, the way how the rough material for natural selection to work with is actually produced, has traditionally been, you know, understudied. And this now could be called as, you know, could be seen as the arrival of the fittest. How is the variation that finally has to be consolidated into this box, how is that produced? And this now has become the territory of the emerging field of evo-devo. And of course, development is important for understanding evolution. Of course, development, we heard a lot about it today, is the process by which single-celled eggs, you know, fertilized egg, transforms itself along the conveyor belt, this black box of development, into a complete organism. And if we then see development, you know, the process of development as sort of, you know, a conveyor belt during which different developmental programs run into sequence to bring it all together, then those programs in one species, they run in a highly predictable fashion, but an important realization is that in different species, those programs run differently. And then if we're taking, a diverse group such as fishes, we have all these differently looking adults, but it's an important realization to just think about that they all will start from this very similar single-celled egg cell at the beginning, and that it is during this process of development where different programs run just in slightly different ways that in the end give a different outcome in the adult morphology. So changes in the developmental mechanisms are the cause, are the root cause of morphological evolution. So if we want to understand the how question of evolution, we inevitably end up with the developmental mechanisms. Now, going back to this box, so I pointed out that this box, it contains the how question of evolution, which genetic mechanisms changed to make organisms look differently. But it can also inform much more on this third box about how we interpret morphology than sometimes appreciated. So if we're looking beyond the species level at diversity, then it has become clear that evolution is in a way predictable. Evolution often, finds the same endpoints in different lineages, while conversely, there are actually constraints, there are evolutionary phenotypes that for some reason seem to be forbidden, that nature's, that the endless beautiful is not as endless as sometimes presumed. And those traditional evolutionary biology has actually had troubles explaining those kind of processes. And just to give you an example, if we're looking at convergence, that the repeated endpoints of evolution whereby similar phenotypes appear independently in different lineages. Here you see, for instance, of course, you all know the hedgehog, which has the nice spikes as defense mechanisms, which occurred multiple times in different mammalian lineages. Of course, an explanation for this is it is adaptive, it's a protective mechanism, but then adaptation on its own, fitness elevating mechanism doesn't seem to be sufficient for convergence, because if we're looking at an example like a frog, it's important to realize 20% of the vertebrates, 1 out of 5 vertebrates, is a frog. It's very successful, yet this phenotype seems to have remained an evolutionary accident, as it occurred only a single time. Then on the other side, we have morphological constraints. So we already heard about the neck, that independent of whether mammals have a long neck like a giraffe or a very short neck like a whale or a dolphin, it's composed only of 7 cervical vertebrae. And it is very difficult to explain what the pure adaptive advantage would be for that. So how does this first box, the mechanisms by which this heritable natural variation are created, how could it inform on this box of adaptive change? And a very important realization here is that to be consolidated as an adaptive morphology in this box by natural selection, you first have to be here. If you don't occur here, if developmental mechanisms somehow are, not capable of producing the variation, that would be beneficial when seen by natural selection, but it never occurs, it cannot end up in this box. At the same time, going back to this convergence, if developmental mechanisms show a bias, a trend to produce a certain type of variation, it will be much more often, offered to natural selection to act upon, and the chances for it to be present at the right time and right place under the right ecological conditions are much higher. So the patterns of morphology in this third box, we can interpret both as an adaptation, something that is favored by natural selection, and this is probably always true, but they are also a reflection of what is actually happening in this first box and how developmental mechanisms can actually produce variation to offer this as the rough material to natural selection. And this is sort of the interface of the questions around which our projects revolve, basically to understand natural diversity, phenotypes occurring in nature, both as a manifestation of adaptation, because most of the time they will be an adaptation, they will be a defense mechanism or something like that, but they also reflect the operation and the capacity of the underlying developmental mechanisms to actually generate variation to give to natural selection. So in this story about this fish, I would like to discuss how these kind of reflections affect the interpretation of this specific phenotype of the bony plates. So you might already have seen it if you like fish, if you like aquarium. So this is a skeletal stain of this species. It's a South American catfish. It's an armored catfish of the genus Corridorus. And as I said, the species doesn't have scales. It has its skin covered in those very strong bony plates. And having bony plates is a very interesting phenotype. It's very fascinating because there are few modern lineages that actually have bony plates. In the past, it used to be much more common. Here you see a nice AI generation of an extinct jawless fish. So if we're speaking 400, 500 million years ago, the first fishes, they actually often had these large, armored plates, which are outward from the outward side, very similar to those bony plates we see in those modern fish. But typically, over evolutionary time, those were replaced in fishes by the kind of lightweight scales that we have now. So those scales in modern fish, they typically are less ossified. You see that they don't form plates, you all know this. So basically they form like little dots that overlap on the skin. And the idea behind is that natural selection prioritized agility, being lightweight, over having heavy protective armor. And the idea is that a phenotype like this, those bony plates, they might have been a very good defense against early predators in the early seas, just by being as strong as possible, but that over time, changing of predatorial strategies, etc., basically an arms race, favored the more agile fish. You would probably could compare it to a human arms race that a knight in full body armor was very effective on the medieval battlefield. It's heavy, it holds you down, but at the same time, it protects you from swords and arrows. In a modern battlefield situation, it would only hold you down. Still, some fish, and those are few, because you have to think that fish, they represent about half of vertebrate diversity. They have gone on and they have lost those scales, and they have somehow replaced them with bony plates. And there's only 3 lineages in the modern fish where this has happened. So typically it's the lineage of the seahorses and related pipefish. There is the South American armored catfishes, and then the lineage of the sticklebacks has done this. And a very intriguing point, because of course, scales and those bony plates, they look in a way very similar. Both are manifestations of the dermal skeleton, is the trajectory by which this sort of change happened. And this is work from my previous colleagues in Geneva, the lab of Pan Montoya. And what they showed is that bony plates structurally evolved from naked skin instead of from scales. So to go to bony plates as a diverse thing, it seems you first need to lose your scales, live for a couple of years with naked skin, and then what reappears are bony plates. So, of course, this is a very interesting trajectory in the context of if we're trying to understand how diversity comes about. So, questions we could ask is, why the consistent pattern of naked skin to bony plate transition? Is this purely adaptive, or does it signify something else? Perhaps sort of a constraint compromise? And why do bony plates then not directly evolve from scales? Is this a developmental incompatibility? Is this impossible, or is something else going on? So what we did is we wanted to investigate this in a comparative approach, looking at scales in an ordinary fish, in comparison with how this is happening in a South American catfish. So a question to ask is, how do developmental mechanisms behind scales and bony plates, how much similarity is this, and where do they differ? So if we're first looking at how this happens in an ordinary fish, we do this work in African cichlid fish. They're actually quite good for this because they have a rapid development, and the scales, they appear on the skin within a couple of days. So you can see here from 9 to 13 days post-fertilization, you get little, perhaps visible here. Then they start to develop in the midline, and then sort of as a wave, they spread over the skin, moving more anterior, moving more posterior, and expanding dorsally and ventrally. And in the end, you get this very nice lattice, this very nice pattern of typical scales on the skin of the fish. And if you would, show this wave in a diagram, then they would start here at the blue, and then the red ends of the arrows indicate the direction in which it is spreading over the skin. So this developmental mechanism, this spreading sort of in a wave, is not unique to fish. So those scales, you saw them sort of when they already start growing, but they derive from a structure called the dermal placout, and the dermal placout is the first indication of a scale, also of a hair, of a feather, or of a scale, you know, in a reptile. So those structures seem to share a very deeply homologous core, and they rely on a self-organizing or reaction diffusion or Turing network. And sort of how this works is during the temporospatial occurrence of those structures on the skin, what you have is an interaction between two signal transduction pathways in which one acts as an activator. In this case, it seems to be always an FDF molecule, which activates the structure itself. So basically, where you have FDF expression, you have a dot, and you will get a scale or feather or hair. And at the same time, it activates an inhibitor of this process. And basically, this inhibitor will then specify, and in this case, the BMP, bone morphogenetic protein signaling, will specify the interplacoidal space, so the spacing in between the different elements. And this process has been known to work in almost all dermal appendages. So we looked at this in our species, if this is actually the same. So here you have in situ hybridization results. So you have FGF20, you have BMP6. BMP6 is just one of the members that is actually doing this. We found 6 members of this gene family that do this independently. And what you see is you have expression of FGF20 here. So you have this first forming row of scales, which then expands to 2, one more dorsal, one more ventral row. And you see that FGF20 structurally precedes sort of BMP6, so suggesting indeed it is upstream. And then what happens, the inhibitor function, if you then see, look in this blue box where BMP6 is highly expressed, you see that actually the FDF that became activated here becomes downregulated. So indeed, it seems that also in our fish, there is a very good evidence for this loop to function. You have FGF20 that you need to form the scale, but at the same time, it activates a BMP molecule that then specifies the space in between the scales. Now, we investigated this mechanism further. One of the kind of experiments we can do very well in our model species is what we call chemical genetics. So basically, we can inhibit by giving pharmaceuticals. We just add them to the water, we can inhibit the signal transduction pathways. So we did a lot of experiments for different pathways. The one that I want to share with you, the most interesting, of course, is what happens when you inhibit BMP signaling. So here you see the expression again of FGF20. So you're looking at the side of the fish, you see here, the forming placoats. Here it already became downregulated in the middle. And this fish has been treated for a while, not during the entire formation pattern of scale formation, but for a while it has been treated with a BMP inhibitor. And 2 things are clear. So the one thing is that here you see the downregulation of the FGF20 in the middle. So it was there, we know it was there. At a later stage, you get progression, it's no longer there. If we inhibit BMP, you seem to lose this repressive action. So that's one thing that happens. And the second thing is that the shape changes. So instead of having dots along the midline, along the dorsal-ventral axis, so here you have a placoat, there you have a placoat, and here you have the scales, separate scales that came from those placoats, you actually get stripes. You get placoats that seems to lose, or you get structures that seem to lose the interplacoidal space, but only along the dorsal-ventral axis. Because along the anterior-posterior axis, there are still stripes in between where the forming scale structures don't extend. So we have this repressive loop. And one important realization from this experiment was that the self-organizing pattern, whereby the FDF BMP loop specifies the interplacoidal space, seems only to work along the dorsal-ventral axis. So at the moment, we have good evidence that actually for the anterior-posterior axis, it relies on a completely different mechanisms, namely on the natural segmentation of the trunk and the division of the trunks in myomeres and myosepta, whereby the forming scales seem to initially appear over the myomeres and where the boundary in between them along the anterior-posterior axis depends on the intervening myosepta. Now, we have those fish. So you saw the expression data. What happens when we grow them a little further? So this is the wild-type fish, so it has the individual scales. And if we then look at the BMP inhibited fish, then you see that those scales, they basically have formed bony plates, or very typically the scutes looking very, very similar to the scutes in the catfish. And also what we counted them, of course, and of course all those scutes, they adhere to the somatic, numbers. So there's a meristic, coincidence of number of those bony plates and the somites, which is also incidentally the same in those developing catfish embryos. So it's a very, obvious question now to ask, do those bony plates in the catfish, do they actually form due to, perhaps an inactive BMP feedback loop? Are they basically scales that transformed into those kind of scutes through this mechanism? As you might now think, it might not be so simple, because I already pointed out that those bony plates did not directly develop or evolve from scales, but that they actually, that there was a period of scalelessness. So to point this out, this is the radiation of the catfishes. Here's the family, based on the family. So this point here, the origin of the catfish radiation, is around 100 to 120 million years ago. And basically the scales were lost here, and most catfishes still preserve this phenotype of being a big, scaleless, smooth-skinned fish. And bony plates only evolved in those restricted lineages. And the interesting thing is that in between the point of scale loss and the first appearance of bony plates that we know of, there was around 30 million years. So there would have been 30 million years of absence of scales. So we had to look a little bit more in a comparative way. So there's the sequence of scale development in an ordinary fish, which, by the way, is very much conserved amongst all modern fishes. So we looked at the sequence of appearance of those bony plates in those catfishes, which is very similar. It starts in the posterior midline. They start appearing more anterior, more posterior. And then instead of forming new rows more dorsally and ventrally, they simply expand more dorsally and ventrally. We looked at a number of marker genes. Probably CTNNB1 beta-catenin is the most relevant one, which also shows that there is, there's homologous expression. We haven't found an expression signature that would contradict homology between them. And then, of course, the most important one is what happened to the levels of BMP signaling. So we would hypothesize that there is reduced levels of BMP signaling or a partially faulty loop of the BMP FGF repression. So how we did this in first instance, we looked at RNA-seq data. Basically, we sequenced the skin of developing larvae of our cichlid fish, shown in green, and of our Corydorus catfish. Indeed, we find overall lower levels, and particularly, we'll come back to this later, we couldn't identify any transcripts of BMP6 and BMP7b. So you saw BMP6 is in the cichlids very highly expressed. It's actually one of the places where it is most prominent. So there are overall reduced levels of BMP signaling during Corydorus skin development. And if we then look at how those genes are expressed, so we looked at several genes. Here you have a Corydorus developing embryos, and here you have the different pieces of the skin here shown in fluorescence, where you actually have the scutes developing. Then if we look at the expression of FGF, we see indeed that, consistent with what we would expect, the entire length of the scute is labeled with FGF20. Those are pretty big fish, so the in situ hybridization, speaking from a technical point, the in situ hybridizations on the Bertoni, on the cichlid embryos, they're pretty easy. These experiments are more challenging. So FGF20 follows the pattern that we expect. And then looking at BMP4, we see that the gene is expressed. But what we typically see in other species is that when BMPs come up, they repress and remove the expression of FGF20, while here the genes during development seem to be largely overlapping, indicating that this repressive loop from BMP4 or any other BMP on the FGFs likely is no longer functional. Something happened in the catfishes that this repressive loop, at least in this context, doesn't work anymore. So coming back to this expression graph, so what happened here? So what we saw is that there is no expression of BMP6, no expression of BMP7b. We looked at those genes in the genome of our fish, or the most closely related fish that was available in the database. And indeed, those genes, they're fully absent. They basically degenerated, they disappeared. Now, of course, we needed to check this in other catfish, so how common is this? And of course, what we first thought is like, okay, this is a smoking gun associated with the appearance, the evolution of bony plates. Actually, the story was much more complicated, but also much more interesting. Here you see a a phylogenetic tree of the catfishes ordered on the family level. And every lineage indicated here that is not black, so either whether it is red or orange, has lost BMP6 in at least one of the genera. And each of the red arrowheads here basically is an independent loss of BMP6. BMP7, it's also not straightforward. BMP7 is similar, although less extreme. It got lost 2 times independently. And how do we interpret this genetic data together with this apparent faulty feedback loop of BMP on FGF? How we see this is that the catfishes, they inherited this loop probably after the initial loss of scales. So the genes were present here, but the scales became lost, and afterwards those genes, they basically lost their function in the process that they had originally evolved from. So what you would get then is you get gene decay. So I haven't discussed this yet, but you might have heard about Dolo's law of irreversibility. It deals with the constraint of re-evolution of lost characters. And Louis Dolo, he was a French anatomist working in the 19th century. And he stated that an organism never returns exactly to a former state, even if it finds itself placed in conditions of existence identical to those in which it has previously lived. It always keeps some trace of the intermediate stages through which it has passed. So, of course, with current day knowledge about genetics, we can interpret this as the use it or lose it rule of evolution. In absence of purifying selection, genetic pathways decay because of random mutations. Even if, an original causative mutation that caused the loss of an organ were to be restored, the network that is downstream is probably gone partially. So if you try in this context to remake scales, then this might not work. So there is a legacy developmental constraint on re-evolution. And to put this back to the context of the bony plates, we interpret bony plates as re-evolved scales. We don't know the mechanism by which they originally got lost. We don't know the mechanism by which they were restored. Catfish, those armored catfish, they have skin denticles that might actually provide growth factors to reactivate the program. But when the mineralization program in the skin became reactivated, the downstream pathway, the downstream self-organizing network was absent. And instead of following a self-organizing pattern, those scutes only rely on the use of a somatic pre-pattern. So the evolution of scutes is highly influenced by a legacy development on re-evolution. So coming back to the slides, bony plates, they follow this trajectory. They evolved from naked skin, not from scale. And if we then trying to answer these questions, why we see this consistent pattern of naked skin to bony plate transition, is this purely adaptive? Are the bony plates simply the best, evolutionary solution that the animal could come up with? Or should we interpret them sort of as a compromise solution? A balance between natural selection and what the developmental mechanisms were actually capable of providing as variation. And why would bony plates not evolve directly from scales? So we can actually produce bony plates by treating a normally scaled fish with a signal transduction pathway inhibitor. Why does this not happen in nature during evolution? To sort of try to answer these questions, so we have an ordinary fish, and in an ordinary fish, self-organizing networks with activating FGF and inhibitory BMP specifies along the dorsal ventral axis, specifies the separation of the scales. So we clearly show with the BMP inhibition experiments that along this axis, the interplacoidal space, for this you need BMP signaling. Along the anterior-posterior space, it seems to be that the pre-pattern at least is redundantly at work. Now, what seems to happen in those catfish, in the scutes, probably those scutes, so they adhere to this somatic pre-pattern, and there's no dorsal ventral spacing anymore because this loop has broken down and they entirely rely on a more simple pre-pattern. So if we look at the trajectory of how we go from scales to scutes, we came up with using this metaphor of the adaptive landscape. So you might be familiar with it. It's a metaphor showing animals on the peaks of a landscape, and the idea is that most species, they are situated on peaks, which are the places in the landscape with the highest fitness, and they can migrate by evolutionary transitions from one place to another place, but they cannot go down into the valleys because that would be forbidden, because this represents a fitness value. So the idea is that a direct transition from scales to skews probably doesn't work during evolution because it seems to defeat the original purpose of the scales, namely to be lightweight and to be flexible. Fish with scales, they already have a protected integument. Fusing those scales probably wouldn't enhance protection much, but at the same time limit mobility to go there. But if you then put in between a phase of naked skin, fishes that lost the selective pressure to have a mineralized integument, evolved a naked skin, and then suddenly are faced with selective pressures to come up again with a mineralized skin, they probably find their way back to scales blocked by genetic erosion of the underlying networks. And the only way to go forward and mineralize their skin is basically to rely on the pre-pattern, and the outcome would be bony plates. So to sum this up in how we interpret those phenotypes, so we interpret the evolution of those bony plates as an adaptive solution, indeed, a mineralized integument, but also as being the reflection of a legacy constraint, the initial the initial loss of the scales and the decay of the underlying genetic network. So if we put it together in those boxes, so we have bony plates partially as adaptive plate, but they're strongly influenced by how the heritable natural variation would have been created, because it's much more likely that in this box the natural variation created relies on this pre-pattern because of the decay of the self-organizing network that normally specifies the scales. Good. Acknowledgement slide. All this work was done at the University of Konstanz, and here the people who are involved. Many thanks.
Thank you so much, Joost.